Hello, everyone. I'm very excited to be here. Thank you to the organizers for setting up this winter school and for inviting me. I come from Paris, from École Normale Supérieure at PSL University, and we are going to have a class about population dynamics. Again, as Akshit was saying, I'm going to use the board. So I'm going to wait 20 seconds. If anybody wants to come closer because the board is small, please come sit in front. No, sorry. No, this is fine. Everything is fine. Yeah, thank you. Okay, very good. So let's get started. I would like to start with some history. Do you know who is this person? I cannot hear it. Somebody shouts at me the name. Leeuwenhoek, yes, this is Anthony van Leeuwenhoek. He's the discoverer of microbes, right? So this is a Dutchman in the 1400s, if I remember correctly, and this man was at the time very fascinated about the small world, and at that time people were starting fabricating lenses, and he bought some lenses and started to look at different samples of tissue, of whatever he can take from the mouth of people, but the lenses were not perfect at that time, and he was working he had just a clothes shop, so he was selling clothes and so, but in his workshop he started to polish his own lenses, and he made the most perfect lenses in the world at that time. And he continued sampling things, and then he discovered little cells, little wee beasts, he called it, which are microbes, and this was the first man to observe them. And he started describing them and sending letters to the Royal Society explaining what it is. So we go a little bit forward in time, and then people were starting to study the microbes and culture them, and so, and then comes Spallanzani. So this is Lazzaro Spallanzani, who made a very influential contribution. He set up this experiment in which he started by boiling some flasks with some soap, or like not soap, but some gravy or some medium where we know that microbes can grow. And he will start by boiling it in different conditions. Some of them were sealed and some of them were not sealed. And then he wait, after boiling, he wait, and he saw that some microbes were growing in the one that was not sealed. And if it opened, then it will also observe that microbes were proliferating. Do you know what question he was trying to answer? Sorry, a little bit. How are they coming into the flask? Okay, and okay. What do they require to survive? What do they require to survive? Okay, but if I was studying how they survive, I will start by putting them in good conditions and then trying to challenge to see if they die, right? But here they are not dying, but they are emerging. Correct. Spontaneous generation. That was at the time a big theory because people were finding microbes everywhere, and they thought the same about worms, for example. They see that wherever there was no worms before, you just leave rotten meat and they will grow. And the moment they start knowing about microbes, they see that they grow everywhere, right? And so they think that they grow spontaneously from nothing, right? And this, to us, is obvious that it's not how it works, but at the time, they didn't know it. They just see microbes coming around everywhere, in meat, in water, in wherever, right? So with this, he made this solid contribution against the spontaneous generation theory, because he saw that if we boil the medium and we seal it, then microbes will not grow until we open. So when we open it, microbes will come from the air or whatever. And the question was not solved here, because then there were some supporters of the spontaneous generation theory that were saying, oh, but you didn't show that you don't need access to fresh air so that spontaneous generation happens, right? And this will take some more time to solve, but we're not going into this. Where I want to go after the contribution against spontaneous generation is the two questions that appear after that, right? Or the two main questions that remain after his experiment. One is, okay, so cells or individuals don't generate spontaneously. So how did the first individual originate? Okay, and this is related to research on the origins of life. And it's a really exciting research direction that is very active right now. But in this school, we are not going into that one. We're going into the second question, okay? Is what is the fate of populations of individuals that reproduce, okay? So we see in biological populations that reproduction happens again and again. So if we have one cell of microbe under favorable environmental conditions, it will reproduce and will get 2 microbes, and they will reproduce and they will get 4 microbes. In essence, the process is the same if we are talking about mussels that also reproduce in a lake or in the water, or if we talk about elk populations, okay? They are reproducing, and everything is about reproducing and trying to survive, as you were mentioned before. Very good. So despite that the populations in biology can be very, very different from each other, you can think that a prokaryotic bacteria has not much to do with eukaryotics like elks. In ecology, we look for regularities, for patterns that are valid for all the populations or across the populations despite their difference, okay? And here I'm just showing some of those differences. So here, if we think about bacteria, they are very small. They are about 1 micron in size. And the generation time is very fast. Under good conditions, in 30 minutes or 1 hour in the lab, bacteria are reproducing, and also their abundances can be very, very high. In a flask, in 1 milliliter of culture, you can get up to 10 to the 9 cells. So imagine the number of bacteria on Earth. It's like, I cannot figure out in my head what does it mean. Then we have eukaryotes of different forms. Mussels, for example, they are visible already, right? So maybe 10 centimeters or so. The generation time is more or less a year. And their abundance can still be pretty high. So in a square meter of a good environment for the mussels, you might find like 10 to the 4 mussels. And only in the Great Lakes, you can find trillions of them. And then for elks, the situation is much different from the other two. So these are bigger animals, and then the generation time might be about 6 or 8 years, maybe 10. And their abundance in the populations are much lower. So in a state like Utah, you might find 10 to the 4 individuals, but in a given forest or in a given ecosystem, you might find just a few couples of them or a few hundreds. Yes? Sorry, can you speak up a little louder, please? The meaning of abundance?
saying the unit.
The units, yeah, okay. The unit, no, this is just to have a sense of what they are, but the units are not the same. So in bacteria, we measured it by unit of volume. The mussels will not grow anywhere in the volume of the sea or of the lake, but just on the surface of the rocks. So that's why I put square meters. And then for elks, I'm just also putting them in the space, right? So in a state like Utah or something. But yeah, one given population of elks might be a few dozens or a few hundreds in a given forest. Okay, it is okay. Very good. And with this, I will just give you a summary of what we are going to work on today. So I will, yes.
It's about the population versus their size. So the size is 10 to the -1 meter.
The size of the individual, sorry.
Individual.
Yes.
So in 1 meter, I can have of the order of 100, but your population is 10 to the 4.
That's right. So it might be that that mussels like the zebra mussel are much smaller than what I said. Maybe they are very small. I'm happy to look into this later and have a chat about this. Sure. But the data comes from paper, so they can actually be very abundant. Okay, maybe the rock has also more surface than we think, but still, I agree with you. And just one last thing that I forgot is that another different thing is how many individuals can come from a unique individual in a single reproduction cycle. So for cells, one cell will give rise to 2 cells, and similar order of magnitude for the elks. But for mussels, one mussel can put at once as much as 1 million eggs, right? So the way these populations behave when you go to the details can be very different, but in the end, they are just reproducing in one way or another. From one cell or individual, you will get a few cells or individuals when they reproduce. Very good. So what we are covering today is a minimal model for population growth and reproduction. We will see that that model is exponential growth that Abby has introduced already in the first lecture. Then we will look for a minimal model for growth saturation. So we know that exponential growth cannot go forever just because of limited resources of space. We will also look into other forms of growth saturation and also other forms of growth dynamics, such as cooperative growth, when cells or individuals need to work together in order to improve the reproduction process. And on the way, I'd like you to present microcosms or microbial populations in the lab as good model systems in ecology that are very convenient for having feedback between theoretical hypotheses and experimental tests. Okay. All right. So with this, excuse me, could we just switch the? There's one thing I wanted to say is what we are not going to include in this class because we don't have time for everything. You will see it later appear there, but basically we are not covering discrete time. You did a little bit of this with Abby just a moment ago. We leave out the discrete populations and population heterogeneity. So here we treat all the individuals as if they were having the same features, okay? We also don't cover any stochasticity. We know that stochastic processes are relevant in biology and in ecology, but here we are going through the minimal. We take the average of the different processes and we work with that, okay? It's just the first introduction, the minimal, we can assume about our ecological systems. And these populations will be well mixed, so there are no spatial effects or things like that. So, as Abby was introducing before, maybe the simplest way of modeling growth is by assuming that the change in the population size or the change in abundances, which is x, is proportional to the number, or to this abundance, right? To the number of individuals that, if I have 2 individuals, they will reproduce at some rate r, okay? So x is going to be our abundance, and r is going to be our growth rate. So if this is the only thing, yes? Okay, should I restart or I can? Okay, very good. So we have that the change in the abundance is going to be proportional to the growth rate of the abundance, okay? And it's important to remember that this is the net growth rate. So this R is the net effect of birth events minus death events, okay? So we will have that R. Keep in mind that is this combination of birth minus death event. So how fast this population will grow? Then we can just solve this equation, and we get something like this. So the growth is just exponential. You will just flip x comes here, dt comes there, you do the integral, and you will get into this, okay? Very good. So again, we are considering deterministic dynamics, as I mentioned before. And despite the strong assumptions that we are making, that everyone is reproducing at the same rate, etc., this model can capture the growth of many different biological populations. Sorry?
Is the probability of having, like, how do you define birth and death rates?
So here you will take just the average rate at which an individual reproduces, and this death will be the average rate at that individuals die, right? So you combine all the causes of death, like high temperature, time to time, or like a famine or whatever. All of those will give you an average rate of death, and this is what you will include. The net will be the growth rate. So it's important to see that this growth rate can now be positive or negative, right? So if it's positive, then the population will increase, and that's why we talk about growth. But with the same model, you can also have an exponential approach to extinction. If R is negative, right? It will go towards death. Very good. All right. So despite all the assumptions here, this reproduces quite well what we see in in the growth of many populations when they are in favorable conditions. So what is favorable conditions for a microbe? It can be a test tube with the right resources, okay? And this is a very simple experiment to do, the most simple experiment to do in microbiology. You just prepare that medium. You have the resources there with the different carbon sources and nitrogen, etc. And then you introduce a small number of cells and you wait, and you will see that they reproduce over time. Okay, so if you take data on how they are growing, this is data that I took during my experiments a few years ago as a postdoc. And we have Corynebacterium ammoniagenes, one bacterial species, growing. So here are 6 different replicates of how it grows in the lab, right? So here we have the biomass over time. And if this linear vertical scale, we change it by a log scale, then we can really see that there is a phase where these populations are growing exponentially, right? So during the first at least 4 or 5 hours, it looks like this growth is a straight line, which is an indication of exponential growth. Okay, and here we can just, yes? Yes, exactly. So we just look at the optical density, so how much light is diffracted or reflected by the bacteria. You have a camera on the other side, and you see how much light has been transmitted. If there are no cells, almost all your light goes to the other side. So this is a way to measure in the lab the biomass, because the more opaque, the more cells you have in, and you can measure this conversion to transform it into how much cells I have there. But take it as a proxy for the biomass, okay? Very good, and we can compute here the growth rate. This will be just the slope of that line, and also the doubling time. So how do we compute the doubling time? Nobody? No contributions? What can I mean about doubling time? Okay, and how can we calculate that? Great, perfect. Exactly. So if what I want is to compute the time at which the population doubles, take the XDT, which will be twice x0, okay? And this is equal to RT. Yes, so from here, this goes like that, 2 equal to this, and then you can have just all right. So if we do this, sorry, yes. So we can do that after we have well, it doesn't matter, but what? Exactly. So T is log 2 over R. Sorry about sloppiness. And that way, we can compute how much time our population takes to double. And for that particular population, it will be 1.3 hours. So every 1.3 hours, we have twice the amount of bacteria that we had before. Let's move into a different biological population. And here, I would like to talk about the zebra mussel. Why did I choose this one? Because it's an invading species, and invasions are nowadays quite relevant and important because they can threat many ecosystems. There are nowadays very frequent invasions by humans transporting accidentally maybe species to other ecosystems. And when they find this ecosystem where they are not native, they can perhaps find a favorable environment in which they don't have competition with their peers from the same population because there is no other cells or individual from that population. If the conditions are favorable, they can grow. And this can lead to consequences for that local ecosystem. In the case of this zebra mussel, which is very, very invasive in the North American lakes, it can reduce the phytoplankton biomass. It can also increase the water clarity because the phytoplankton goes down. This has a consequence on the growth of plants, and then the snail population that will eat those plants. So just the introduction of one species can really introduce heavy, dramatic changes in different ecosystems, right? So because of that, having a model of how these populations grow and try to predict how much it will take to get to these important changes can help us having a sense of what are some strategies that we can take, or how fast we need to act in order to stop all of that, right? So scientists have been monitoring the growth of the zebra mussel over the last 20 years in lakes from North America. And one thing to notice is that, again, if you just look at linear scale, for a long while, you will not even notice that zebrafish are there. But one of the properties of exponential growth that we can see here through plotting in log scale is that this exponential growth in linear scale will just rapidly jump to high abundances. So this looks like the previous year, there were no zebra mussels, almost no zebra mussels in my lake, but now it's full of them, up to 10,000 mussels per square meter, okay? And we go to a very different population, the population of elks. They have been reintroduced. So this is another kind of invasion, but it's a desired uncontrolled invasions. Elks had been hunted to almost extinction or to extinction in different parks and in natural parks in America. And in recent times, they have reintroduced them to restore the ecosystem somehow. And in that sense, here we are seeing the estimated population of elks after reintroduction in 3 different regions. So in the Lake Huron North Shore, in a park in Wisconsin, and in Bancroft. And what we can see is that these elks, after introduction, they also grow exponentially, even if it's at a rate that is much lower than we see for the other populations. These were the successful introductions, but they can also fail. So in some other cases, they reintroduced elks in the park, and the death rate was higher than the birth rate in those environments, and the introduced elks went to extinction.
But the elks are dying, you don't expect exponentially.
Probably not, because we are also at a very low numbers, and then also stochasticity will happen, and also
finite lifetime collect.
Correct. When
it's astonishing what you do. 0.5 orders of magnitude.
But they take a population of people in this
Not people, bacteria.
like eggs, they eggs of different ages, right? If they're distributed across all the ages, then each one will have, imagine each egg lives to be, I don't know, 14, and then they would die exponentially.
So not exponential, there'll be a very significant break connection.
If they all have the same age, yes. But if they're across all the different ages, then they would die exponentially.
Not exponential.
Okay, I will have to say that on average, if we look at different extinctions of elks across different ecosystems, we take the average is maybe exponential, maybe it's not. But if you look at one single extinction, I will say that because of stochasticity of how many die every year, that will really not.
But what I'm getting at it, just a larger problem. So you see a lot of exponential increase.
Yes.
Very few data about good exponential decrease.
Okay.
Of animals. But nevertheless, exponential, I think what people should get out of this is the exponential part, at least for increase, is robust, but it doesn't mean that this model is what's causing it. It's something much more general about eigenvalue of a big complex system with heavier linear mass equation, something like that. So what do we take away from the fact of exponential growth? It's not that these equations are correct that lead to exponential growth, but something.
Okay. Yeah. It's new to me, and I'm happy to chat a little bit more about it later. Very good.
I have a question.
Yes.
So in bacterial population, it's easy to see if it's exponential or not.
Yes.
But say in elk's population, you don't have that many individuals. So when you see birth rate or you see like death rate, there are like really discrete points, and you can fit any graph like to it, exponential, linear, or something. Then how would you conclude that if it is exponential or not?
Maybe the way to think about it is that depending on the level of detail that you want, exponential growth can give you a good description of it. It's not going to be perfect, it's not going to be exact, but here they don't have a lot of individuals, but still you can see how this increase is linear over the measurements that they have been taking. Will that answer your question? And it's not really.
Like say if I see for a long time and they have like good resources, and then it is exponential, but like it's not going to happen, and you will always have like discrete points, so it will never be exponential. Like you can't conclude it that it's exponential. Like if
so okay, is your question about the discreteness of
discrete? If it is like too discrete, then you can't conclude what type of growth it is.
Well, again, we are doing huge assumptions here that are not true. Of course, if you really want to model the behavior of them, you will take into account more details such as the discreteness of the reproduction events, etc. You may conclude that it's not exponential, but maybe this is also due to other factors, as you say. If they are not in a very favorable environment, it might not be. But in principle, I will say that if they are growing after reintroduction, here exponential growth is giving us a decent explanation of how they are growing. Maybe it's not conclusive, but it's a minimal model is the first thing we do. And then if we are interested in more details, then we add some more complexity into our model. Okay.
Sorry, I'm just wondering, I'm asking clarification. Are you sort of visualizing more like, okay, if we have these data points, let's say for population at discrete time points, how are we drawing a line through it? Is that what you're asking? So that can also be a statistical or like a curve fitting exercise, right? And so you can say which curve or which form fits this data the best. And do you know if that's is the exponential fitting it best or something else? Maybe, does that help, like to get at whether this is the correct curve or not?
When we fit data points, we like say this is the equation and vary these parameters and then fit it. So we are choosing equation, like we are assuming it's exponential. So if we choose exponential, quadratic, and linear for the starting, everything is gonna fit.
No, so if it's a statistical exercise, for example, everything won't fit the data as well. If you have this spread of points, everything won't fit the data
Yeah.
as well. There might be one that fits it better than that's something that we test in statistical framework. You see, like here, we can when we put the points on the board, and then we see that the log of the population size gets described by a straight line. So averse is something else. That should be a statistical exercise. We start off to get at what the pattern.
Okay.
This is the values of R which you get from these curves,
Right?
Is there any estimate of the individual values of B and D as well? I mean, is it that B and D are both significantly larger than R, and just the difference happens to be small? Is that the case, or do we know individual B's and D's?
So I don't know them. I imagine that they have it, because if they were reintroduced in a national park, it's very likely that they were monitored, maybe even with some data that like emitter or some some device that the scientists can track. I don't have the answer of what they did with them. I will say that birth and death rates might not be very high here, because like if we start with just a few dozen elks and they reproduce once a year or so, and they don't reproduce much individuals at a time, each couple, I think that it will be low for both death and birth.
I think like a very basic idea of like B would be how many options you have divided by your life. So how many options you have in your lifetime, that is the number B, say over 70 years, that is your B, right? And D is 1 over your lifespan. And it's a personal approximation, right? So if you were just to type old age, so X to I every year.
Yeah, thank you for the contributions. Keep interrupting whenever you want. Of course.
Can I just quickly add to that? I think that was a great question, and perhaps even speaks to the one of the lectures that we would be doing in terms of theory data integration. There are field methods, and which closely link to even things like state structure population models, in which you can, like field ecologists can go out and get estimates of birth and death rates from collaring individuals, tracking them over time, looking at how the population, like how many new ones are on average born. So there are methods to do this, and they do go into, particularly for populations of concern such as this. It's not always possible because it's a huge logistical challenge for many populations, but it is done and it can be done.
So let's continue. And as you might have noticed, this exponential growth doesn't last forever, right? At some point, the populations are gonna run out of resources. In the test tube, for example, they just consume all the carbon source that we put in at the beginning of the experiment, and mussels must just colonize the whole surface of the rock, so there's no more space, or there's no more nutrients for growth. Okay, so eventually this exponential growth has to slow down, and the next thing for us to do is to try to model that growth saturation. And
while counting helps to count every single element, there's something on the German tank experiment that you count a few of them and you get an estimate of the entire set.
Sorry,
while counting elks, do you count every single elk to get the population, or?
In the end, it's an estimate, because they might not be sure that they have counted all the elks, but yeah, they just do a few counts during a short amount of time, and probably they average, or they just estimate based on the error they make how many they might have. So it's not a precise measure in that sense. Very good. So populations might run out of resources, but there are also many other things that can limit growth. For example, every biological individual from any species will be secreting something out of their metabolism, right? So we are secreting some things that can turn into toxins. We will not like to live there. The same happens to microbes. They consume carbon, and in the metabolic process of getting some energy, they are releasing protons, they are releasing different things that can be toxic. So eventually they cannot keep growing in that environment. Maybe there are still resources, but there are also many toxins that microbes produce. So there are very different reasons to stop growth. And perhaps if we want to model it, and Abby introduced also this simple or minimal model that we can take. In this sense, we had our exponential growth as before, and perhaps the easiest thing to assume is that the decay of this is a linear function. So, sorry, this is x dot or dx dt. Let me write it all in the same way. And a very informative way of thinking about this dynamics of time is to think about the per capita growth rate. The per capita growth rate is quite universal. We can define it for any kind of growth equation. It's just how is the change divided by the number of individuals that we have. So on average is how much one individual, or how rapidly one individual is growing or dying, producing offspring or dying, right? So this gamma here is the per capita growth rate. And if we plot it, see that this per capita growth rate, if we assume this minimal model in which the individuals are competing with themselves, so they are lowering the growth rate of the population, as we increase the abundance, this per capita growth rate is going down, and it's going down linearly. We are assuming that the competition between individuals of the same population is just proportional to how many competitors do you have. And this will go down and it will hit 0. So the growth rate will be 0 and the population will be stable when we reach this K value, which we call the carrying capacity. Okay, so I'm gonna write it here. So if I start here, where will it move the population? Right or left? Let's say window or wall, right? So if I start with this many individuals, we have a positive per capita growth rate. So the population is growing, and then we move in that direction. What happens if we start here? The opposite, right? So everything above, everything on the right hand of this side will give us negative growth. And exactly at the carrying capacity, we have a stable point. There is another stable point in logistic growth, which is when x is 0, okay? But in this case, we see that this is an unstable point. So if we start at 0, there are 0 individuals, no one reproduces. But as soon as we have a few individuals, then they will grow, and this growth will start decaying until we reach the carrying capacity. Okay. Very good. So here is how this means a stable point, fixed point, and when I draw it like this, will be a stable. So what did I wrote here? Yeah, fixed point. So this is an unstable fixed point, and the other one is a stable fixed point. Okay. So I didn't realize that I have covered the first part of this exercise that we are going to do now, but maybe you can work on these other two for just a few minutes, and then we will continue. Yes. Previous graph.
Just wanted to understand why in the case of the muscles, for example, you see a lot of fluctuations about the modeled equation early on, but once you hit the constant region, you don't see as many fluctuations. Is this just a result of this data, or is this something that you generally see?
I'm not an expert in field data for mussels, for example, so I couldn't tell you. But what I can tell you is that across microbial species, this is very variable. So I presented this one because it's very reproducible, but I could show you microbes that grow more or less exponentially and then they die, and other ones that's doing funny things. Okay, so it can be very different.
Is gamma like a reproduction rate of individual?
Sorry, what was the question?
Is gamma like a reproduction rate of individual?
Is gamma is the per capita growth rate. So if here this was the per capita growth rate, which means just divide by the abundance, by x, the equation that you have for the dynamics of the system.
No,
right?
as in x is the number of population, right? Like how many individuals are there, and dx dt is the rate. Right.
I'm not sure I'm understanding the question. Sorry.
So dx dt is the rate of growth.
This is the rate of growth per capita, which means for an individual, this will be the growth rate of one unit of the population. No.
What I mean is dx dt is the growth.
Yes, so it's the change in the abundance over time.
Yeah.
Right. No, so the change in abundance at a given time for the population.
Yeah. And x is the number of individual at that moment.
Yes. Yeah. Exactly.
Yeah.
You can take
if I divide it like dx dt by x, what does that imply? Growth rate of individual.
Yes.
So that is like how much one individual is giving to the system.
Exactly. So if
that's
I
reproduction.
exactly. So if I have an individual, how much that individual is growing or dying. Okay. And when you plot this one, it will change with the population number. So even if you have the same per capita growth rate as in here, the value of this will be much larger if you are at higher abundances or lower abundances. But when you look at what's happening at the scale of the individual, then here you will have the same per capita growth rate. Okay, which is not true in the logistic model, because as you go to higher abundances, then you start competing with your peers, and at higher abundances, the per capita growth rate, so this measure of the fitness of the individual, if you want, goes down.
Mm-hmm.
So, does anybody has a sketch for these time trajectories? So if we start, for example, at very low abundance, how does this look like? Exactly. And should I continue? All right. Right. Okay. So as the growth, as the population is reaching higher abundance, this growth rate has to go down, right? And then it will become zero the moment we reach this carrying capacity. So starting from low abundance, this has a sigmoidal shape like this, okay? If we start like this, it will be very similar. We will just reach carrying capacity a little bit earlier, and earlier, okay? What if I start above this carrying capacity? It goes down, right? If I start above the carrying capacity, then this term becomes negative, so we have a death rate, so an exponential approach to extinction. This is how the time course in the logistic model looks like. Okay? And so what happens if we harvest this population? Harvesting is like hunting, if you want. So what we are going to do is to add a death rate to this model, okay? And then let me do it here. We add this term minus delta x, okay? So our equation is now dx dt equals rx 1 minus x over k minus delta x.
Delta of x or delta x?
Sorry, delta times x. So how will this carrying capacity change here? Or better than the carrying capacity, how will the stable abundance will change? And a very easy way to analyze this is to retake this diagram and add what will be the death rate. This death rate is constant with the value of x. So we can think about it like this death rate that we have to subtract to the actual value of the per capita growth rate, right? So in this case, if I just subtract this to the logistic growth rate that we have, these lines now cross at 0 at this point. So now the stable abundance will be here in the case of harvesting. Okay, I'm gonna indicate it with H. Is this clear? Have you seen this kind of analysis before? Any questions? Why is it stable? We saw here before then that for values that are below the carrying capacity, we grow, and here for larger values we will die, right? So if we subtract this death rate, the actual value will be, the new value will be 0 here. Then if I go left, the logistic growth rate from before is higher than the death rate, which is here. And then this difference will be positive, so we'll still go to the right, and vice versa.
Just on that point, not only pedantic again, but the growth rate is assumed as a positive number. The R is assumed positive, and sort of the death contribution is also assumed positive, and this difference of the two. So when the 1 minus x over k is negative, you have death rate, which makes up the difference. And that's why you have a leftward movement on the x-axis. So would that be a reasonable way to understand this model?
Yes, I missed
what. So what would this mean when, for example, x is greater than k? And it means there's a death rate.
Yes, exactly. So if correct, it's if we were mentioning this before. So if x is larger than k, then this becomes negative, and then the evolution. In terms of your r is equal to b minus d, and if I overlap this on top of that, it means d is 0 below k. Correct. And increasing above. That's sort of the. Okay. Yeah. So. Or is it when it's a fraction of that described bird? Then how would you ask people to interpret the negative part of that?
Like easy species, right? Imagine we have the fish would die by itself, but then we catch the fish, so it dies faster, right? And so it's an extra death. It's like fish. So there's a D naught, which is dependent on nutrient limitation, and there's plus D, which depends on harvesting. 6 years, but because in fish, when it's 1 year old, then it dies.
And finally, just to conclude this part, as Abby was introducing before, we can also plot the population size as a function of this death rate, and we are assuming that the growth rate is larger than 0, okay? And in this case, what you have is that the population size, the stable population size, will go down linearly until it reaches critical death rate. Okay? Very good. And this death rate has to be R. Right. So could we, excuse me, can we switch to The intercepts are different.
Section diagram or the phase?
The intersections are different. With the, sorry. Sorry. Do you mind? Why don't you come and draw what you mean? Just
when we were plotting this, the carrying capacity.
Yes,
so when
delta, you see that the slope is -1, right? But then there's an intercept which is that first if you don't know r times 1 - so, okay. So that intercept is what will be will be what you have for delta equals 0. That's the carrying capacity with delta equals 0, and which we just keep.
And up to here, we were assuming that the population, the saturation of the growth is linear. Okay, but there's also no reason to believe that this is the case. For example, it could be the case that as long as we have enough resources, so more than a threshold, we don't compete too much, so everybody is able to grow at their maximum growth rate, while as soon as we reach some limit, some threshold of resources, then we start experiencing the death rate, right? And there are also other ways that populations could interact with, and they could also interact in a less competitive way. So adding 4 competitors will not be just double the competition that I feel when I have 2 competitors. Okay, and in that case, let me. What we can do is to extend our logistic model to capture other forms of growth saturation. Okay, so in this case, what we will have is that correct. Yes. Yes. So we can extend our model just having an exponent that will capture different forms of competition with your peers. Okay. We just rescaled by this because then it's a way of being able to capture different kinds of model, but this is not very important. Also, if it goes away if we have theta equals 1. When theta equals 1, we have the logistic model.
So the idea here, Danny, that the theta is the main shape of how this how portrait behaves with population size.
Correct. Yes. Yes. So, for example, if we have our linear decay so if we have the logistic growth here, then with this, you could have behaviors that are like this and behaviors that are like this. Okay? Just by changing the theta. We are not going into much details on this because of time constraints, but I'll just...
What sort of mechanistic interpretation of theta? And it's also top.
What is sorry. Yeah, so what is modeling the competition here is this minus x over k, right? Because the larger the value of the abundance x, then the lower the per capita growth rate, which means that if there are more individuals of the population, the reproduction rate goes down. Okay, we have assumed the minimal case before, that is that this competition is linear. Oh. So if I have 2 individuals, I have a level of competition. If I have 4 individuals, I have twice this level of competition. This doesn't need to be the case.
This is the competition between a species, like intraspecific.
Exactly, between the species. So if you take the per capita growth rate, so we divide by x, right? This will be the fitness of an individual of the population. And as we have more competitors in our population, so more cells in the case of the test tube with microbes, then this will go down because of that competition, whatever is the origin of the competition.
And there's no practical manner to infer theta. It's something we...
Here, we are not, no, exactly. So here we are just saying that we are assuming that it's linear, but it could be not linear. And then if we can extend this model, but assuming that this goes with a different exponent instead of an exponent of 1.
No, what I was looking for was that for R, we can, we can give a mechanistic interpretation, and for K also we can give that. For example, K could be the area or the resource limitation of such sort. For theta, like what would be a mechanistic interpretation of some sort?
So that's the thing, that it might depend on the value, and it might not always be, there might not always be an explanation that is generic, right? It can be because of different factors.
The kind of competition that can that
which kind of competition?
Different types of competitions, the different types of competitions that we know of. I'm sorry, I forgot which competition, the particular definitions of the ones. Can that tell us something about the value of the theta as such, predict, or the resource distributions?
My sense is that there's no generic answer, that you will always know that this kind of competition gives you that exponent. But then,
if we can think about the kind of competition that is going on in between the individuals of that particular population, can we predict how R is going to change with K as such? And can that help us with a mechanistic understanding of theta?
Did you say, sorry, you asked about how the competition is changing? Did you say with K or with what? With theta, yeah. Yes. So
the nature of the competition.
I mean, in a way, yes, because if theta is negative, for example, this will be sublinear. So you know that as the population grows, the relative contribution of the competition with this growth is smaller than will be by just adding up the individual contributions as in the logistic model, right? But it doesn't tell you if that competition is for a resource or if it's competition for space or for any other thing. Is that what you are trying to get at?
Yeah, I think also microscopically, maybe you're asking if you can figure out what organisms are really competing.
Or like when you are making a model, not like empirical data. If we have empirical data, maybe we can get data from there. If we assume the sort of competition that is going on between the individuals, I'm not being able to change the question.
Yeah, I think this is the kind of
the type of competition that is going on between them. The types of competition that we know of, like how the individuals are competing.
Yeah, I think like it makes sense that like if you're taking competition for resource, right? To make a new individual with many resources, right? So it would be the linear operation that you see in the model, it's very natural. But then if you had something like big, right? When you're crowding, I eat less, and I go with other ones, right? Or I'm stressed out, that's why I learn, right? Or other things, those would appear as deviations from the thing. So any social behavior of like the competitors or even like changes in the outlook, these things like trying to form say that they can find out how many there are around them. Those would all be compounded. One parameter that they become really dispute.
And just writing a few things because of time constraints, but sorry, let me do something. So again, the value of theta is not very informative of the mechanism of the competition, but what we can do is to write the form of the equation depending on the value of theta, and then we will see that there are different equations that we get. So, for example, for tumor growth, this equation captures in many cases well the growth of the tumor, right? And then the explanation that the scientists gave is that this growth is limited by the blood supply into the tumor, and this blood supply maybe reaches with a higher efficiency the surface, the external part of the tumor, but maybe it doesn't reach all the way in, and then this changes the competition with the amount of cells, right? And it happens to be that it's well captured by this model. Then we can have a sublinear competition as well, which means that the relative competition with your peers is smaller than in the logistic growth. And then scientists have found that there are many different biological populations that will differ from this logistic growth. And in some cases, they are captured by the theta logistic model. As I mentioned, for the growth of some tumors, this theta logistic model with theta equals to 0 is is working well, but it's not always the case. The growth of the tumor can be behave like a power law, for example, for these different kinds of cancer, like breast, lung, or rectal cancer. Here, the scientists have measured the volume of this tumor and the total lesion activity, which is a measure of also of the change or the growth of this tumor. And in that case, it follows well a power law model like this one. And recently, there has been some evidence as well about biological populations that are across different species and across different ecosystems. They might not be growing logistically, but they were well modeled by a sublinear growth of this kind. Here, they were measuring this slope k, which translates to theta as k minus 1. And then we are just talking about populations dynamics here, but later on, we will get into competition with other species and so. And observing this kind of sublinear growth has also some consequences for the way different populations compete or they react to competition, and how many species can coexist and live together in a stable way, for example. We are, again, not getting into details. It's just to show you that we can go beyond the minimal model of logistic growth to capture different forms of growth in biology. And just during these last 10 minutes, I would like to switch to something different, that it's not the saturation of the growth, but the form of growth that is not, that it doesn't have to be just competitive. We know that some populations cooperate in order to survive or in order to grow. In the case of prey, for example, like fishes or zebras, it becomes effective to pack and be together to kind of escape the predators in different ways. In other cases, we know that for sexual reproduction, for individuals that have males and females, for populations that have males and females, if the abundance of the population goes down a lot, then it might start being very difficult to find mates. So in that sense, the per capita growth rate is very low when there are not many individuals, but as soon as the population is increasing, the per capita growth rate becomes higher or more effective.
Just clarify the use of the word exponential and linear, sub-exponential, sublinear. Right? I mean, these words are very confusing. Here you're using the word superlinear growth, but you really mean supra-exponential growth. Is that right?
Yes.
Is that? I mean, because if beta is greater than 1, then it's faster than exponential.
Yes.
And here, what you mean is that beta is less than 1, so it's sub-exponential.
Yes, beta has changed across the different papers. It means something different. Okay, so here beta is just the exponent of the power law. Here, beta is what they are able to measure, which relates to this theta exponent there as k minus 1. Okay, but so this k, if we get so if we subtract the 1, we will be in the region with theta is negative.
So I'm a bit confused about those two equations there. The theta was the power to which 1 minus x by k was raised, whereas in the second equation it is the power to which x by k is raised in the bracket. These two are not the same mathematical form.
You are correct. Sorry?
I think it's more common than that.
So, okay, so maybe, let me check, but it could be.
This one is only where the other one is a bit more complicated than ours.
Right. Let me check if I made a mistake. I did make a mistake. Sorry. So the actual formula is like this one. Theta tends to 0, but it's not 0.
How did that the first one become a log function? One minus, yeah.
This magnitude H piece, but in the log case, we're not allowing external. I can understand X being carried.
If
so, you can never end
correct.
up with no cells.
Yeah.
You can, right.
Then you have X point for different reasons.
Exactly, yeah. But also we are modeling tumor growth, so I don't.
But I'm still a bit confused a bit here. Do you mean theta less than 1 or theta less than 0? Theta less than 0 has a singularity.
I mean theta less than 1, correct. Between, sorry, yes. So it's sublinear when it is minus.
You mean 0 less
Right,
than
so
theta less than
if it's
1?
no, but I agree with.
Yeah, I
It
think
is.
you can have theta negative. It's just that for x equals 0, you deal with that sort of separately, right? So x equals 0 is always going to be ex equals 0 for everything else.
So is
no, even yeah, even and also maybe.
So,
Maybe then if you
yeah, please go ahead.
just go back to what Sanjay was saying, I think it was maybe an important point. So I'll reiterate. Yeah, I think there can be something confusing when we're talking about growth here and calling it sublinear or super exponential. And so we need to be a little bit careful about what exactly we're talking about, right? So there's a few things that we can talk about. One of them is just how the whole population is growing, right? And so the whole population can grow either exponentially. For that case on the left, that would be when beta equals 1, or when beta is greater than 1, it could grow super exponentially. That would be the population growing. But then there's also this other thing that we often talk about, which is how the per capita growth rate, which is not how the whole population grows, behaves with the population size, right? So that's some function. And so that function can either be linear or sublinear or something else. So yeah, there's a bit of a confusion here because lots of people will say sublinear growth, but they're not talking about sublinear growth as in the growth of the population. They're talking about how the per capita growth rate function behaves with the population size. So yeah, this is an important point to bring up. I think that's why maybe I stole a minute to talk about it.
That's right. And in that case, when we talk about sublinear, we're thinking about this per capita growth rate, the saturation of this per capita growth rate.
About the population itself, I suppose, in that case.
In sub-exponential growth?
I mean, and that I presume would be the situation where beta is less than 1. This beta is less than 1.
This beta is no, it's so okay. So that's the theta is between -1 and 0.
So theta is something that is characterizing the growth rate in those formulae there. Whereas when you're talking of sub-exponential growth, are you talking of the growth rate, or are you talking of the population itself?
I would say about the growth rate, but what do you mean about the? No, sorry, the population itself will grows. Right. No, I think what we are talking here about is about sub-exponential growth saturation. So still about the sorry. Okay, I see. Okay, I see the confusion. If I can just change this, which was wrongly written, I will say that this is sublinear growth saturation. Okay, so what we are talking about is that in the logistic growth, this per capita growth rate we will have theta equals 1. The growth saturation, which is this term here, is linear on x. And with theta negative between -1 and 0, this will be
less than 1.
Then
Between 0 and 1.
Okay.
Paper is a bit like different because they don't put like the what was the quadratic part to change. They changed the linear part to something. So they don't. They leave like the logistic part. Like the quadratic part is quadratic, but what used to be linear now is something. These 24.
I see.
It's equivalent to this kind of.
Okay, so this correct. This is how they can measure is so over an extension of the, and so it's not per capita in that sense, but just it's a measure of density of the population anyway, right? Because this is the way that data is accessible as well. Okay. So I agree with you that this for B sublinear, this should be between 0 and 1. I'll take a look at what did I do with my lecture notes, and maybe we can chat about it later, or I'll mention it tomorrow. I wanted to mention one more section, but given that it's already the end, maybe I can start tomorrow the lecture with cooperative growth and then move into interaction between different populations. Okay. Great. So far as a concluding remark today, we have seen the minimal model for growth, which is exponential growth. And then we know that because of competition by limited resources or other reasons, we can have growth saturation. We need to have some form of growth saturation. The minimal model for that is the logistic equation, which assumes that this saturation is linear with the abundance of the population, but there are other forms in which the growth can saturate as well in real populations. We can model some of those, maybe for sure not all of them, but some of them can be well captured by the theta logistic model. In the case of the sublinear, this theta should be between smaller than 1. Okay, and we're going to leave it here, and tomorrow we will move into how populations can grow cooperatively, and then how populations can interact when we have different species in the ecosystem. Okay? Thank you very much for your attention.